Finish our chapter today, though. So this last part, 15 or 16 slides or so, and kind of it stresses the importance of how transcription and translation are done. So let's start with DNA. Okay, so we'll say we have a DNA sequence which is TAG, CATTACG. 1, 2, 3, 4, 5. That's a T. 1, 2, 3, 4, 6, 7, 8, 9, 10. We need two more. Let's move this out of the way. We'll do C. All right, that's our DNA coding sequence. What's our RNA going to be? A, U, C. Okay. Next. U, A, All right, that's our mRNA. Okay, and we'll say that is our mRNA that's been edited, spliced. That is the coding sequence completely. Okay. Now, what are the anticodons going to be in the tRNA? U. Okay. Now, when you are reading codons, if I gave you the codon chart, we're reading codons, not anticodons. So we would be reading AUC, GUA, AUG, CGG. And in your book, somewhere in the slides, you can look up, take the couple minutes right now, couple seconds, and look up those codons. So flip over to the slides, the little chart you have, or in the book, and we'll figure out what those codons are. So AUC, when you get it, is which codon? Which codes for which amino acid? So this is isoleucine, what we do, ILE. All right, which GUA? Alanine, ALA. What? Valine, all right. That's okay. Valine, okay, VAL. What's AUG? It's methionine. Okay, start codon or methionine. Okay, and what's CGG? Arginine, ARG. Okay, so we have three of those. All right. Now, that is your amino acid sequence, and that is important for the production of a functional protein. Okay? So now, what happens if the ribosome wasn't started at the right point? What happens if we move the ribosome over one? So instead of starting at the A, we're going to start at the U. What is this then? This codon, UCG. What amino acid is that? Serine. SER. UAA. That's a stop. What happens? What's happened? You end up with what we call a truncated or a non-functional protein. Okay? That is an example of what we call a frame shift mutation. The reading frame, where the ribosome is going to change or read codons, has been shifted. Okay? So now when you shift it over, that causes a problem. Okay? What happens if we shift it over? Oh, let's say we shift it over a little bit more. So instead of starting at the U, we're just going to skip 3. And we're going to start at GUA. Now what happens? So we still get valine, methionine, and arginine, right? We just skipped this first one, correct? So, because of the triplet code, multiples of 3 are important. So if the ribosome starts 1 over, that could cause problems. If it starts 2 over, that could cause problems. But if it starts 3 over, it might cause a problem depending upon where the start codon is, or it could just mean one amino acid is gone. All right? That might not be that bad. So we have what we call frame shift mutations. Some of them are called nonsense mutations that introduces a stop codon early. Or we have missense mutations. Missense mutations means that not the right amino acids are going to be put in. So just by sheer good fortune, I picked out of my head this DNA sequence, and it just showed up that it actually worked out the right way. That we have a frame shift mutation. It introduces a stop codon, which is a nonsense mutation. If I had a missense mutation, it might be say we start at CGU. That could be introduce 3 different amino acids, and that would be a missense mutation. Okay. The point being is that you're going to change the amino acid sequence completely. Changing the sequence of an amino acid, of a polypeptide, changes its structure, which ultimately may change its function. Good or bad. Okay? What I mean, good or bad means advantageous to the cell or disadvantageous to the cell. Okay? Advantageous means maybe it has a growth advantage over other cells, and that could actually drive mutation, all right, of organisms. All right, it could be disadvantaged, and the cell could die, which also has important for evolutionary consequences. All right, so let's look at the slides. Let's minimize that. So we have mutations, the change in the genetic material. I've talked about changes in frame shift mutations. We could also have a point mutation. Okay, point mutations changes. We're going to change the way that one amino acid or one nucleotide of that codon. Okay? So if we go back here and we look at valine here. Okay? What if I change valine, or I change the codon for valine, which is GUA. Let's change that first codon. Let's say there's a problem here. Instead of the G, for whatever reason here, we end up with a C. Okay? So the codon is actually CUA instead of GUA, which means it would be what amino acid? Leucine, LEU. Okay? That is a point mutation. We're changing one particular nucleotide within the sequence. And that can happen at the DNA level or at the mRNA level. Okay? Most likely, it's at the DNA level during problems with replication or in changes in the DNA sequence. Okay? But that is a point mutation. And it will effectively change one amino acid. Examples. Sickle cell anemia is a point mutation which changes one amino acid in hemoglobin and definitively disrupts its function. That is a worst case scenario. Most of the time, changes in point mutations are not that bad. Okay? And especially point mutations in that third nucleotide of the codon, they can be masked. All right? Meaning that because of the wobble effect, the fact that that last codon can code for the same amino acids, that change might not lead to a change in the protein. Okay? They could be evident or they could actually be hidden. All right? So you have a single nucleotide in DNA template strand could lead to the production of abnormal protein, but the disruptive. This is your hemoglobin example. All right, you're changing from one, you're going from a glucine to a valine. All right, but what that does is that causes sickle cell hemoglobin. It changes the way hemoglobin, or the ability of hemoglobin to bind and transport oxygen. Okay, other types of small scale point mutations. All right, two basic categories. We just talked about a nucleotide pair substitution. We're going to change one nucleotide pair. We're going to change the DNA sequence, so we're going to alter it in front, and so it's, instead of TAA, it's going to be AAA, whatever it might be. All right, and then other things which are more important, okay, more disruptive, is an insertion or a deletion. Okay, so if we have the normal DNA sequence, which is A, T, A, C, G, G, A, right? A nuclear paired substitution would we're going to take out, say, this A, and we're going to change it to a T. Still the same number of nucleotides, right? Would code for the same number of codons. It's just going to be different codons. The more disruptive ones are when we move and we're going to add bases to it. So instead of we're going to A, T, A, right? And then we're going to add, say, CG to it. And then we're going to, let's add some more to that. We're going to add CG, A, T, and now we're going to pick up the CG, G, A. More proteins, right? More amino acids in the protein, correct? What does it do to the frame shift? So I've added one, right? So this would, here would be, eventually be one codon, right? If we were to transcribe this. This would be another codon. That's a whole added amino acid. And now because I have this one added, now I've changed the reading frame of that DNA or of the RNA from that. All right, so if we did it out and if we transcribe this DNA, this would be U, A, U, G, C, U, A, G, C, oop, that's a C, C, ooh, that's pretty good, U, U. Gosh, can't write with this thing. All right, so if we looked at the codon, what's UAU? Anyone still on that page? TYR, tyrosine. What's GCU? Alanine, okay. What's AGC? Okay, and then what's CUU? What is that? Leucine. Okay. Now, how is that different from the what would be UAA would be tyrosine, right, for this one. And now CGG was what? I think that's alanine. Arginine, CGG, and then AA, we'll just call it AA.
Wouldn't that be?
It would be UU, we'll just call it UUG.
Wouldn't the second one be GCC?
Oh, I'm sorry, yes, GCC. Yes, you are right. So this second one is GCC, alanine, right? I'm reading DNA over here. Alanine. And then UUC. What's UUC? Serine. All right? Different. Okay? So what I've done, just by, in this case, luck, I've actually added and shifted the amino acids, right? But just by luck, I haven't changed tyrosine, alanine, serine. Okay? Just by perchance. Okay? So I've added one, but I haven't changed anything until the end. Now, say if this fourth one was a stop codon, say if over here the next codon was a stop codon in our original DNA sequence, that would mean the end of the protein. But in this case, I've added one. Maybe it's leucine and then a stop codon. Maybe that's not that bad. All right? But typically, these insertions or deletions, they change the reading frame, and that is where you end up with a lot of really what we call deleterious mutations. All right? And this is showing your nucleotide pair substitution, T instead of a C, right? What it does at the end, the important part of this is at the end. So there's your template strand, your mRNA, and the protein normal, okay? You have 4 amino acids and then the stop codon, okay? You introduce one mutation, in this case, at the end, it's a silent mutation, all right? So in the first box you see a silent mutation. Because that mutation, that point mutation doesn't change the amino acid, there's no point, there's no result in changing the protein. It's silent. Okay, so it doesn't really do anything. You change the first amino acid of that serine, or the glycine. You change it, you end up changing the glycine to serine. That's a missense mutation. You're changing the normal amino acid sequence to a different amino acid sequence. And that could lead to a change in shape, a change in function of the protein. A nonsense mutation, at the end, you're introducing a stop codon before normal. Okay? A nonsense mutation results in a truncated or a shortened protein sequence, a shortened protein or amino acid sequence. And then you have insertions or deletions on the right, okay? You have the frame shift. Insertions or deletions are going to change where the ribosome is they're going to change the order of the codons, how they're going to be read. In this case, it introduces a nonsense, a truncated protein, right? Second case, an extensive missense. Now the protein is going to be the same, and they're going to change the stop codon. It's not going to be, could be too long, could be lots of different proteins messed up. All right, and then in this case, there's a deletion. There's not a frame shift, right? But one amino acid is taken out, and it could be one or more amino acids are taken out, resulting in usually a dysfunctional protein. Okay, as an example, so. Plenty of mutations that can occur. All right, and we just went through these, so I'm going to go through these quick. Nuclear pair, silent mutations, just said what they are, right? And we can go through each of them, need to, because we just went through them. Missense and nonsense. Missense means different amino acids. So the codon, your original codon, whatever it might be, right, has been changed and now results in a different amino acid from the normal or the original protein. Okay? A nonsense mutation means that it introduces a stop codon before normal. All right? Earlier than normal. Okay? And it usually leads to a non-functional protein. Okay? Finally, you have your frame shift, insertions, deletions, your frame shift. Frame shift changes where the codons are read and can lead to really, really bad changes in the protein. All right, and that comes in either nonsense or missense mutations, or it could lead to amino acids. Each of these are set up in a box that. Okay, where do those mutations come from? Well, consistently, constantly, every day, your body is exposed to mutagens. Substances that will cause DNA or mutations, okay? Whether it be the air we breathe, the coffee we drink, the water we drink, whatever it might be. Right? We are constantly being exposed and subjected to mutagens. Right? They're always there. All right, but anything, and they could, spontaneously occur during DNA replication, recombination, repair, you name it, all right? But a mutagen is anything that can cause a mutation in DNA. And then eventually those mutations can cause changes in protein or mRNA, and the codons, eventually into protein, right? And there's different ways that we've developed tests to do that, and practically all cancer-causing chemicals, carcinogens, are mutagenic, right? They are mutagenic in that they disrupt, they cause mutations, and that whether or not, for the most part, a lot of times, that causes a change in a gene related to cell growth, and that leads to cancer. All right? So, in review, we started transcription translation with the idea of what a gene is. All right? So we know a gene codes for a protein. It is heritable, okay? It's a part of the DNA chromosome, and it's passed on from one generation of cells to the next, right, through the DNA replication and codes. That's the genetic code for a functional protein. Usually, one gene codes for one protein. Well, we know that's not necessarily true because of alternative splicing, right? Know what splicing is? It's still one protein, right, polypeptide, and that can be changed, right? So, your overall definition, gene can be defined as a region of DNA that can be expressed to produce a final functional product. That kind of takes into consideration the effect of alternative splicing. A gene will produce a final functional product or many functional products, right? Either a peptide or an RNA molecule, right? At first, the idea of a gene was one gene, one polypeptide. But now, we know that some RNAs are produced and they're not translated, but they are functional, such as ribozymes, right? Those RNAs that are important for splicing, ribosomal RNAs that are important for the function of the ribosome, transfer RNAs, right? Movement of amino acids. Those all have functionality, but they are not proteins. So, and they are genes, coded for genes within the DNA code as well. So you end up with this definition, right, which covers both polypeptides, RNA molecules, and also encompasses the products of alternative splicing. Questions on transcription, translation prior to your exam on Friday? Now your review sheet is posted. Anything else? If you have any other questions, we will go on from there.